Okay, hello everybody. So this is the video. Sorry, I'm like setting myself up in position. So this is the video for membranes in transport. So you are going to hear me recall information about biomolecules here. We will talk a little bit about energy and ATP and expand on both of those things. So here we go. Let's jump in. Okay, so these are some of the videos that I recommend to help you put all of this together. Once you are done with lecture and you've taken your notes, maybe you come back after a couple of days and you look at these videos to help refresh your memory over what this lecture video covered. So in our last video over cells, we talked about a lot of different membranes. And we can take all of those membranes and kind of categorize them under the big category of biological membranes. So here are some things you have to remember. Biological membranes are going to have both lipid and protein molecules in them. So we're going back to those macromolecules again. We're going back to the lipids, which are types of fats. We're going back to the proteins, which are made up of amino acids. This is why you can't forget that stuff. Biology builds on things that we've already talked about. It's very foundational. So membranes in a living system are based on what we call a phospholipid bilayer. And that phospholipid bilayer is very fluid. It moves. It can't be too, what's the word I'm looking for here? It can't be too stiff, because if it is too stiff, it'll break apart and float away. And we don't need that. Cells live immersed in liquid, mostly water, so we have to make sure that they can move and change and adapt based on their environment. So being made up of phospholipids and being a bilayer is important to that ability to stay fluid and to stay organized. So the phospholipids themselves make up the bulk of the membrane, and then embedded within those phospholipids in different areas. And this is one of the places where it gets hard to picture, because we're talking about a 3D 360 model. It's not just that circle that we draw, it is encompassing. Remember, cells are spherical, or kind of spherical. Plant cells are more cuboidal, but again, it's a 360 thing. So within that membrane at various points, we have these proteins that are embedded. Some of them go all the way through the membrane. Some of them are only found on specific sides of the membrane. It just depends. Now, for the proteins and the phospholipids to coexist together and to allow the membrane to keep its shape, they have to have certain chemical properties that are very similar. So they are going to have to be hydrophobic. Remember, that means I don't like water. And they're also going to have to be hydrophilic, where I do like water. At least regions or sections of them are. Remember, another word for hydrophobic is nonpolar. Another word for hydrophilic is polar. Okay? So they have to have this dual nature, mostly because the majority of the membrane is made up of phospholipids, and those phospholipids are amphipathic. They like water on one end and don't like water on the other end. They also have to have dual solubility properties, meaning they have to be able to allow certain types of things to come in and out of the membrane, and how they enter the membrane is going to depend a lot on their chemistry. So the polar regions of the phospholipids, the regions that like the water, can be found on the surface of the bilayer. The surface leading to the external environment of the cell, but also the surface leading to the internal environment of the cell. Because don't forget, it's a double layer. Now, in the middle of these two polar regions are the nonpolar regions that we normally refer to as the tails, and they don't like water. They are trying to get as far away from water as possible, so they tuck themselves in the middle of the layer, or layers technically. Okay, so again, for a quick recap, this is what your phospholipid looks like. Remember, that head region is the region that likes water, but that tail region does not. So when they get all arranged, the head regions are always going to be on either end. So all of these heads will be facing the water outside of the cell, while all of these heads are facing the water inside of the cell. Okay, and this is what I meant when I say it's a 3D all encompassing thing. We tend to think of cells as just these little circles that we draw, but remember, cells take up 3D space. So this membrane is found all the way around. So in our biomolecule unit, we mentioned cholesterol, which we sometimes refer to as sterols. And we said that they have very specific roles or functions in how the plasma membrane operates or behaves. So, sterols are found within the membrane, specifically within the nonpolar regions. They're going to be found inside where the tails are, okay? Now, it's not that sterols don't have polar regions, they do. So, let me label that on the drawing real quick for you. But the majority of the molecule is actually hydrophobic. So just that OH on my cholesterol is going to be the hydrophilic region. So when you look at this part of this picture, you can see that all of the little OH groups are associated with the head regions of these phospholipids. Same thing down here. Okay. Now, it means that every other part of this molecule is hydrophobic, which is why the majority of the molecule can be found in between the tails, because that's the area of the bilayer that is also hydrophobic. Okay, go back to my highlighter. So the sterols are going to be found in a nonpolar region, those middle regions, of those hydrophobic tails. But their polar end is going to be found in the area where the hydrophilic heads of the phospholipids are found. Now, cholesterol is the main kind of steroid that's going to be found in animal membranes in particular, and its job is to help keep the membrane fluid. Okay, so what is the main function of the lipid regions of the bilayer? So, like I mentioned before, the phospholipids are going to make up the majority of the bilayer. So, their job is mostly structural. They've set up the structure or the framework of all of the different membranes, be that cell membrane, nuclear membrane, whatever kind of membrane. They serve as a barrier. They're a gate, they're a fence, if you will, that prevents the passage of most water soluble molecules in or out of them. So they control what's going in and what is coming out. So here's a great picture. It's a little dark. Oops, sorry. But it shows a little bit of how these guys are arranged. So here I have my bilayer. I can see all my phospholipids. If you look closely right here, you can see some of those cholesterols embedded within. And then these blue things are going to be proteins. And let's see. So if you look closely, all of these go through the membrane. But like I said before, sometimes they can be found on just one side, depending on the type of protein. So this would be the outside of the cell, and then everything in here would be the inside of the cell. And to be honest, a lot of these proteins that run all the way through or are found on the inner layer are important for anchoring that cytoskeleton that we talked about in our last video. All right, so here's a really good image of what this bilayer looks like when it's completed. Okay, so we have proteins and everybody in there. I'm going to highlight the names of the parts and pieces that are really important for you to make sure that you know. So, let's see. Of course, you need to know plasma membrane. Oh, maybe I should change the color. I'm hoping green will show up. We'll see. Okay, so of course you need to know what the plasma membrane is. You need to know the difference between an integral protein and a peripheral protein. You need to know what a glycolipid is and what's the difference between a glycolipid and just a regular phospholipid. You need to know that these little chains standing on the outside of some of the proteins and some of the phospholipids are actually carbohydrate chains. and that's where the glyco comes from. You need to know cholesterol, what it does and where it's found. And then some of these more specific ones, like here where it talks about gated channel protein, that is important, but we'll talk more about that in depth coming up. You also need to know what a glycoprotein is, what a transport protein is. So notice that we have big categories. Like we can talk about integral proteins and peripheral proteins, but then I have different types of integral proteins. So I have gated channel proteins, I have transport proteins. You need to know what all of those things are and what they mean and what they do. Okay, so let's talk a little bit more about these membrane proteins. So the membrane proteins are suspended within the bilayer individually. They're very rarely like grouped together or attached to each other. That entire blue structure is one protein, okay? So sometimes we use the term mosaic or even fluid mosaic to describe the membranes. So fluid tells me it moves. Mosaic is in reference to being made up of lots of different parts and pieces. So when you look at that bilayer, it has lots of different types of structures within the bilayer itself. So we sometimes use the term mosaic to represent that. So just like with my phospholipids, these proteins are going to have areas that are hydrophilic, that really like water, and they'll be found on the extremities, of the layers. So facing the outside of the cell or facing the inside of the cell. And then they'll also have regions that are hydrophobic, and they will be found in the interior of the membrane. Now, as these proteins stop being efficient at their job as they get older and they're no longer functioning or doing the work that they were designed to do, they get pulled out of the membrane and degraded back into their amino acids, and those amino acids get used elsewhere in the cell, and then a new protein will have replaced that old protein. So, proteins in general are the workhorses of a living system, a cell or an entire organism, if you will. And they have lots of different functions. So, we'll start skipping through those in a minute. But the proteins embedded in the phospholipid bilayer perform the following functions in terms of the majority. So, they transport materials, especially materials that are hydrophilic, across the membrane, so in or out. They help with cell to cell recognition. They help with the reception of signals or hormones, information carrying molecules. They help cells to adhere to each other if they need to perform a function that requires two of them. And they also help with metabolism. So it's important that you know these major functions of membrane proteins. Okay, so starting with the two big categories of integral proteins versus peripheral proteins. So integral proteins are embedded all the way through the bilayer. They're deep in the bilayer. So deep that if you remove them, they without replacing them pretty much immediately, they can cause the layer to disperse. Okay? So the way I need you to think about it is these integral proteins span the entire membrane. So if we look over here, here's an integral protein, and notice how it goes all the way through from the outside of the cell all the way through to the inside of the cell. Every integral protein is going to do that. Now, compared to a peripheral protein, so they are found on just one side of the membrane. They are on a surface. They're either on the outer surface or the inner surface. So this one's a little harder to see with this particular image, but I'll do my best. So right here, this is an integral protein. Whoops. Notice it's only on this inside section. Sorry, this is a peripheral protein. I might have said integral. I meant peripheral. Here's another one, only on the inside layer. Okay? So like I mentioned before, we sometimes use the term fluid mosaic model to describe membranes, and it just means that the membranes themselves are not stagnant. Those phospholipids can move and do move, and they can rotate around themselves. They can switch locations with the phospholipid on their left or on their right. They are mobile. Mosaic, like I mentioned, refers to the fact that we're made up of all these different types of proteins that mostly just kind of float in the lipid bilayer depending on where they're attached or how they are attached. Oops. Oh my goodness. I need to change the settings on my pen apparently. Okay, so now that we've spent a little bit of time talking about what makes up the membrane, let's now focus on the job that this membrane does. So, the major job of the cell membrane or of any membrane in a living system is to help with transportation, moving things in or out of the space that that membrane encloses. So, this can be broken up into two big groups, if you will. We can talk about passive transport and we can also talk about active transport. So when we say passive, we mean no energy, so really no ATP needed. So it's just going to happen naturally, if you will. Okay? So the most common example of passive transport is diffusion, where it's the movement of molecules with a concentration gradient. So what does concentration gradient means? It means that we're moving from areas of high concentration to areas of low concentration. Right, so if we look at the simple sketch, if each of those red dots represents a molecule, then molecules naturally move from where there's a lot of them, so high concentration, to where there are fewer of them, low concentration. So in this situation, these molecules would be moving in this direction, and they would do that until they are balanced on both sides. So that's what with the concentration gradient means. So this does not require any sort of energy because we're just allowing the natural movement of all molecules to let them spread out. Think about if someone enters the room with wearing lots of very strong smelling perfume. Even if you are not sitting close to the door or sitting anywhere close to that person, eventually you will smell their perfume because it just diffuses through the air. It moves from where there's a lot on that person's body to where there's none or very little the rest of the room. Okay? Now, our plasma membrane has a unique feature. We always describe it as being semi-permeable or selectively permeable because it only allows certain substances to pass through and anything else is going to have to get help of some sort. So we'll talk more about that in a second. So when it comes to our cell membrane, passive transport is going to happen through the actual phospholipids of the membrane. It does not involve the proteins at all. Really important for you to know that. And again, it requires no energy. It is passive because we are moving with the concentration gradient. We're moving from high to low concentration. So if you look at the image over there on the right, you can see that for this particular molecule, outside of the cell is where we have high concentration. And inside of the cell is where we have low concentration. And as time passes, those molecules start diffusing straight through the spaces in the phospholipid membrane itself. So not through proteins, but just through the natural spaces in the phospholipids. And then over time, we end up with the same amount on the left as we have on the right, or on the top as we have on the bottom. That's passive or transport, also known as simple diffusion in this case. Now, the smaller the molecule, the faster this process is, especially if those molecules are soluble in lipids. So they have a similar chemistry. They mostly don't like water. Think about it that way. Okay? It's easy for the membrane to allow things that are also hydrophobic through. So, another form of passive transport is known as facilitated diffusion. Facilitated in this case means with help. Who is giving that help? The proteins are giving the help. Diffusion tells me that it's still going to be a passive process, meaning I'm not directly using any energy to make these molecules move. So, why would we need the molecules to pass through proteins? Well, very often it's because they may be the right chemistry, they may have the right chemical makeup, so they're hydrophobic, but maybe they are really big molecules and they cannot fit through the small spaces in those phospholipids. Okay, so with facilitated diffusion, it's going to need help from membrane proteins. It is still going to follow the concentration gradient, so we're still going from high to low, and it can only be utilized for certain substances. Not everything can pass through one of those proteins. So, a great example of this is actually water. Water does not pass through the bilayer because water is a polar molecule. So, right away, it's not going to interact well with those tails. The tails will prevent it from passing. So, water must pass through channel proteins. And an example of a channel protein is this image that you can see right here. And those have specific names. The ones that transport water are called aquaporins. Aqua for water, porin for pore, hole or opening. Okay, so with facilitated diffusion, I am still going to move from high to low concentration. So I can move from outside the cell into the cell, or I can even move from inside of the cell to outside of the cell, just depending on what I am and where I need to go. But I'm still going from high to low concentration. So it's still not costing me any energy in order to do so. A lot of the proteins are going to look kind of like tunnels where they just have a space and the molecule just flows in through that space. Okay, another example of passive transport is through what's known as carrier mediated transport. Carrier mediated transport are carried out by carrier proteins. So, carrier proteins can only bind a specific and single solute, single molecule, and it transports that molecule across the membranes. If you look at the picture, you can see that we pick up the molecule and look at the shape of the binding site. It's very similar to an active site on an enzyme where it has to fit. So it picks it up and then it opens up or closes itself first, actually, so that the molecule can't go backwards. And then it opens the other side and the molecule diffuses through that side and it enters into the membrane. We're still going from high to low concentration. A very special and specific type of passive transport is known as osmosis. And osmosis is the diffusion of water. So we're still moving from high to low concentration, but the only molecule that we are thinking about, or the only molecule moving, is H2O, water molecules. And this happens across a selectively permeable membrane, just like the plasma membrane is. Okay? Very often, water is going to move in response to the change in concentration of other molecules. Okay, salt would be a great example here, or even glucose would be a great example here. So as the concentration of glucose changes or increases, it creates a difference in where is high concentration versus where is low concentration, and that's going to force water to move naturally from high to low concentration. So when it comes to water movement, another thing we have to consider or think about is tonicity. And especially because our cells live their lives surrounded by some kind of solution. Yes, mostly water, but not water alone. Okay? 75% of our body is water, but we also have other things dissolved in that water. And that is going to affect how your cells either pick water up, so water is entering, or lose water. Water is being lost from the cell to the rest of the environment. So with tonicity, we are specifically talking about the amount of solute dissolved in a solution. And while the solute itself may not directly move, increasing or decreasing the solute will affect how water moves. So there are three types of tonicity, if you will, and they are hypotonic solutions, hypertonic solutions, and isotonic solutions. So in hypotonic solutions, these are solutions that have a very low concentration of solute. Okay? So I need you to think about both things here. I need you to think about how much solute there is, so then how much water can there be. You have to think of them both. Even though only water is moving, the solute concentration is going to affect how much water there is to move. So in hypotonic solutions, we have a small amount of solute, which normally means we have a larger amount of water. Then we have hypertonic solutions, which have higher amounts of solute concentration, so less pure water. And then lastly, we have isotonic solutions, which have equal amounts of both water and solute. Now, in hypertonic and hypotonic situations, water is going to move. But in isotonic situations, there is no real obvious final movement of water. Water might move a little bit, but it's an even exchange. So however much I lose, I will also gain back because the concentration is the same on either side. Okay? Excuse me. So if you look at the diagram, sorry, my alarm just went off. So if you look at the diagram, if I take this bag of sucrose solution, which is going to represent my cell, and I put it in pure distilled water, that sucrose solution, because it has the sucrose in it, is going to have less free water than this beaker full of nothing but H2O. Okay? So the beaker is high concentration for water. This bag will be low concentration for water because it has so much sucrose in it. So water is going to move into the bag, and that would cause the bag to swell or to get larger. That's if I put it in a hypotonic condition. If I take the same bag and I put it in a hypertonic condition, so remember that means that this solution is going to have a lot of sucrose in it. The solution has 10 moles of sucrose compared to 2 moles of sucrose. Moles is just a measurement. So if I take this and put it into this environment, now because I only have 2 moles versus 10 moles, I now have more pure water in this bag and less pure water in the beaker. So the bag becomes high concentration, the beaker becomes low concentration, water will move out of the bag, which would cause it to shrink. And then if I take my bag and I put it in the exact same conditions as what's already in the bag, then yes, I'll have a little bit of water come in and a little bit of water come out, but my bag will be unchanged because I'm having what we call net exchange. However much I lose, I'm gaining the same amount back. So if these were cells, and this cell sat in this solution for too long, eventually it would get so big, if it was an animal cell, that it would burst. If it sat in this situation for too long, it would lose so much water that the cell itself would die. It would crenate, would be the word we use there. And then this situation mimics what's happening in our bodies already. Okay, so the concentration of water in our cells is very similar to the concentration of water in just our general bodies. So our cells aren't shrinking or expanding. When you are a nurse and you are hooking someone up to an IV line, understanding these properties are so important, because if you give someone the wrong amount of saline solution, you are creating either this situation or this situation, where what you wanted was this situation. Now, when it comes to plant cells, you have the added feature of a cell wall that has to be taken into consideration. Now, plant cells have this big vacuole in the middle, like we talked about right here. The more full that vacuole is with water, the more that vacuole is pushing on everything outside of it, including all of the organelles, the nucleus, the cell membrane, which then will push on the cell wall. So you can see in this picture, the vacuole is nice and full, which means that this cell has gotten lots of water on the inside. So under hypotonic conditions or situations, plant cells are actually completely full. This is considered normal turgor pressure for them because that vacuole is pushing on everything in the cell, and that allows your plant to stand up nice and straight. Kind of like that. Like I said, I don't draw well. Just go with it. Now, when plant cells are in hypertonic conditions internally, the opposite happens, and this process is known as plasmolysis. So this cell is going to actually lose water to its environment. And when that happens, look at the vacuole. The vacuole has shrunken in size, so it's not pushing on the cell. And if it happens too much, all of the plasma membrane, will start pulling away from the cell wall because all of the cytoplasm has also lost its water. Now, normal turgor pressure in an animal cell would result in that cell bursting. But in a plant cell, that would result in this cell standing up nice and tall, being filled to capacity, so your plants are nice and upright. This plasmolysis situation could lead to your plants completely dying. Now, under isotonic conditions, I still have some turgor pressure. I'm still full, but I'm not completely full in terms of my vacuole. So this is where your plant starts wilting. All right, so it'll look like that. And guys, I apologize if I sound a little nasalier than normal. I'm having an allergic reaction to something, and this is just what I sound like for right now. So I'm so sorry if it makes my voice more annoying than it already is. It's all good. So now looking at the same conditions, but this time in animal cells. So the example we're using here is a red blood cell. So if I take my red blood cell and I stick it in a hypotonic solution, water is going to enter the cell. So water is going in, and over time, that cell will swell. Okay? Now, left too long like this, the cell may even hemolyze or burst. So that is a term that I need you to know. In the opposite situation, if I take my red blood cell and I put it in a hypertonic solution, that cell is going to have more pure water than the surrounding solution. So water will actually leave the cell. It'll diffuse out of the cell. Whoops. Okay, so you can see that cell starts to shrink. It starts to get these weird puckered situations, and that's known as crenation. So when that cell shrivels up, that's called crenation. And in animal cells, because they don't have that added protection of a cell wall, all of these situations can lead to that cell dying. With the plant cell, that cell wall protects it so it won't die. It can be revived, or it takes a longer time for it to die. Now, in an isotonic situation, we have what we call, there's no overall movement of water. So we have gross movement, if you will. I might be losing water to the environment, but I'm also gaining water from the environment. It's an even exchange. So this results in the cell not changing size. There's no shrinking. There's no getting bigger. It's just a normal sized red blood cell. So up to this point, everything that we have talked about has been passive, does not require the use of energy or ATP in any form. We're going to switch that up now, and we're going to start talking about active transport. So active means with energy. We're using ATP. All right, so active transport requires energy, either directly or indirectly, and that is going to come from the hydrolysis of ATP. Right, so remember, hydrolysis is that reaction where we're breaking things up into smaller pieces. We're breaking bonds by adding water. Normally, with passive transport, things move with the concentration gradient from high to low. With active transport, things will be moving against the concentration gradient. So we will be going from low to high concentration. And that's why the cell has to use energy, because we can't go with the flow. Think of it as going uphill all the time. So we're going to utilize some of those membrane proteins that we mentioned before. It's going to be a specific kind of movement depending on the surface. And we can have situations where that molecule or that substance can become saturated at high concentrations. So things can only move so quickly. A classic example of a molecule that utilizes active transport, or molecules, I should say, are the molecules of sodium and potassium. And they actually move through the same protein. So we call the proteins that move them a sodium-potassium pump. Now these pumps move a specific amount of sodium in a specific direction and a specific amount of potassium in a specific direction. So we call it a known exchange of sodium potassium, and you need to know what that is. So we move 3 sodium molecules out of the cell, and for every 3 sodium molecules we move out of the cell, we move 2 potassium molecules into the cell. Okay, so 3 out, 2 in, or 2 in, 3 out, however you want to think about it. So let's look at our diagram that kind of shows this. So notice here in my protein, in my transport protein, I have specific sites that are specific shapes. These grooves are where the molecules are going to enter and exit. Excuse me. So starting with this top picture. Now this is going to be the inside of the cell. This is the outside of the cell. The part that's open has a high affinity for binding sodium. That just means that sodium binds really easily to the molecule when it's facing the inside of the cell. So 3 of those sodium molecules are going to go ahead and attach themselves to those sites, okay, where they fit. Now this same molecule has sites for potassium, but on the inside of the cell, it has a very low affinity for binding sodium. So the sodium binds, as you can see in this picture. We're going to have the breakdown of ATP into ADP, and that gives us the energy for this pump to open up on the cell, the outside of the cell part, and close itself off on the inside of the cell part. Okay, so we call that a conformational change. Now, when this happens, this molecule is going to now have a low affinity for binding sodium. So the sodiums it was carrying is going to leave. They will not rebind, and instead, 2 potassium molecules are going to bind, because now once we're open to the outside of the cell, that pump has a high affinity. It's easy for it to bind potassium. Once the potassium has been bound, we'll go through another conformational change. We'll open back up to the inside of the cell, and those potassium molecules get dumped into the cell. Okay, so that's how ATP is used to get this pump to open and close and bind these 2 different molecules. So remember before I said active transport either requires a direct or indirect energy source. So the sodium-potassium pump is an example of a direct energy source. We have to use ATP on that pump in order for it to work. What's pictured here are examples of indirect energy. So these are known as secondary transporters, or sometimes they're called co-transporters. It's still a form of active transport, but they use an electrochemical gradient that is generated by active transport to transport a second molecule. So it's not going to use a direct energy source such as ATP. It's just going to utilize the fact that we're already moving something, and that's created a gradient that we can take advantage of. So they come in the form of a symport and an antiport. So what is the difference? A symport moves the molecules in the same direction. Okay, so my secondary molecule is going to move in the same direction as the original molecule that is driving the electrochemical gradient. Okay, so here's what I mean by that. So these red molecules are the ones driving the action of this protein. They're naturally going from regions of high concentration, there's lots of red dots, to regions of low concentration, just a few red dots. As they pass through the molecule, enough energy is created indirectly through their own gradient to move these blue molecules as well, which are actually moving from low transport or low concentration, to high concentration. There are only 3 blue molecules here and lots of blue molecules here. So we're going against the gradient, right? So these guys are just naturally flowing, and the movement of them from high to low creates a gradient that allows these guys to move through as well, except from low to high. So that's a symport. Your antiport moves solutes in 2 different directions, opposite direction from the molecule driving the action. So again, we'll step through it. So this time, we're looking at our red molecules who are still our driving force. So we're going from high concentration of red molecules to low concentration of red molecules, but we're going from outside the cell to inside the cell in that direction. That action is going to allow the cell to also move these blue molecules, but the blue molecules are being moved from low concentration to high concentration, which in this situation means from inside the cell to outside of the cell. So several slides ago, we talked about endocytosis, how the cell brings material in. Well, what about waste material? How is the cell getting rid of things that it doesn't want? Or how is the cell getting those proteins that it makes for other parts of the body out of the cell itself? So that process is known as exocytosis. We touched on it briefly, but didn't really explain a whole lot in detail. So exocytosis is the release of molecules outside of the cell, and it happens via those secretory vesicles. Those vesicles work hard, y'all. They have lots of jobs and help lots of things to happen. So a vesicle is just a bag that's pretty much made by phospholipid membranes, and they can hold things on the inside. So they surround the molecule that needs to be transported. You can see this in the picture. And they travel up to the plasma membrane where they fuse with the plasma membrane. They just join it. And then that opens up the quote-unquote bag, and the molecules can then diffuse to whatever part of the system they are needed. So the fusion is just what we call the vesicle membrane joining the plasma membrane. And this act of fusing opens the vesicle so that the contents that it was carrying are now released to that external environment. Okay, and even though we've already talked about endocytosis, we're going to run through that again. So again, endocytosis is how materials get brought into the cell. They come in 2 versions. Pinocytosis, which is cell drinking, normally used for small molecules, and then phagocytosis, which is cell eating, normally reserved for larger molecules. So the molecule gets enclosed in a vesicle that starts off as part of the plasma membrane. That plasma membrane just folds itself, encloses the molecules in it, and then travels through the cell and deposits that molecule wherever it's going. There's also a kind of endocytosis known as receptor-mediated endocytosis, and that works for very specific molecules. So if you look at this picture here, you can see that. So here we have what we call target molecules. Those target molecules will land on proteins on the outside of the cell that we call receptors. When those receptors are filled with their target molecule, kind of like an enzyme, only then will the membrane fold and then carry all of the material inside of the cell. Okay, and then this gives an image to what phagocytosis looks like. So like I said before, phagocytosis is used for large molecules. Like I'm eating a bacteria, I'm eating some kind of big thing that I can use for food. Let's think of it that way. It's almost like a form of this little bacteria is my prey and I'm going to hunt it down, engulf it, and digest it. So the cell has the ability, especially animal cells, have the ability to kind of extend their plasma membrane and then push their cytoplasm up into that space that they've created. It's called cytoplasmic streaming, and this is what's happening here in phagocytosis. So here's my prey. The cell is going to send this section of its cell membrane this way. This section goes this way, so you see that here in this picture. And then it fully encloses the material and pulls it into the cell. So here's a picture of an actual amoeba cell doing that with whatever this material is. All right, that's that for transport and membranes, and I will talk to y'all soon. Bye, guys.